Biology, Chapter 19, Theory Video 10, Analyzing and Storing Genetic Information. The first thing that we are going to discuss is about microarrays. So this is a highly advanced piece of technology, which actually, which is a simple slide of about 2 square centimeters. And on this, there are thousands of tiny spots containing known DNA sequences. So they have the DNA molecules are attached to the slide and act as probes to detect lengths of DNA or RNA with complementary sequences. So obviously, you can understand that microarray would have to contain single strands of DNA. Then these single strands of DNA would join together with complementary sequences of DNA and would go through base pairing to form double-stranded DNA. So this actually gives us an understanding as to how many how similar the DNA, the genes are, and the gene sequences are to one another. So this process of binding together of two molecules of single-stranded DNA by complementary base pairing is known as DNA hybridization. So you can actually compare these genomic DNA between two species as well. So to identify how similar they are, we can use this. So there are probes. There are some things called DNA probes for containing a wide array of genes on this microscopic slide. On this slide, then having microscopic dots. So they actually if there is a genome, if there is a gene present which has complementary base sequences, then they will join together to produce a double-stranded DNA molecule. So then what happens is we can compare them and say that these genes are similar in both of the species, or when we compare whoever or whatever we compare. So to see that they actually join together, so let's say the DNA from species A would be labeled with green tags, fluorescent tags, and then species B would be with red tags. And when when the labeled DNA samples are mixed together to allow for hybridization, they hybridize with the probes on the microarray. So if they don't bind to probes on the microarray, they will be washed away. So then what we need to do is we need to inspect using the ultraviolet light. So this microarray will have all the sequences of DNA in it. So if the samples contain these things, these genes or DNA, then we will know if when they combine. Now, for example, there would be many type of single, the similar gene sequence would be present, or the similar base sequence would be present in many numbers. So not only one. Now, because we are checking A and B, two DNA from two species on a single array, then what happens is if A and B both contain this gene sequence, then the green tag will also join up with the array, that base sequence on the array. So it is something like the millions of samples are there in this, thousands of samples are there on that. So when the samples are complementary to another sample, what we are checking, they will join up together. So something like that. So the green ones will be, if the green gene is complementary, then the gene one will retain. If the red one is there, then it will retain. When if it does not have a color, then obviously that gene is not present in. It is present in the array, but it is not there because array originally had it. So the complementary genes are not there in both of the A and B species. So as a result, it does not have. Um, then when we observe, if both of the species hybridize with a probe, then what happens is a yellow color would be seen. So the yellow spots would show that two species have the DNA with exactly the same base sequence. So if there is no color, we just realized what it is going to be. So you will get a microarray. If you view with it with a laser scanner, you would see see it like this. Then after, so this microarray would be scanned with a computer, and the data would be stored by the computer, and we can compare them, and it will actually give us the data that we require. The microarrays not only make it not allow us to compare the genes, but also allow us to see which genes are expressed at any specific time in the in each cell in the body. If now, for example, in cancer cells, the genes which are expressed are not similar to an active non-cancerous cell. So you can compare the genes by identifying which genes are being transcribed into mRNA. Uh, so again, we are going back in reverse. The mRNA from the two types of cell would be collected, and reverse transcriptase is used to convert mRNA to cDNA. Then the mRNA, because it is number is small, we can by using PCR we can increase the quantity of mRNA, and as a result cDNA. Then this cDNA will be labeled with fluorescent tags and denatured to give a single stranded DNA, and we will allow it to hybridize with the probes on the microarray. So the spots on the microarray with fluorescence would give us that the genes are transcribed in the cell. So the intensity of light emitted by each spot would indicate the level of activity of each gene. If there is a high intensity, that would indicate that there's many mRNA molecules present in the sample. Low intensity would indicate that there were few. So they are not only going to show us that the genes are active, but their level of activity also will be shown using these things. Then to find the mutations of the genes involved in breast cancer also can be found out using this microarray system. Write down a note on this. Analyzing and storing genetic information, microarrays. Microarrays are slides that are printed with thousands of tiny spots containing a known DNA sequence. The DNA molecules attached to each slide act as probes to detect lengths of DNA or RNA with complementary sequences. These thousands of DNA sequences exist in multiple numbers, and when they are allowed to hybridize, within brackets DNA hybridization, binding together of two molecules of single stranded DNA by complementary base pairing, binding together of two molecules of single stranded DNA by complementary base pairing, they will show that each gene or DNA is present in the sample checked with the microarrays. The DNA on the samples being checked would be color tagged, and after hybridization, when they are checked, the presence of colors will indicate that that particular gene is present in the sample, and if two samples are being checked and a combined color is provided, it shows that both of the species would have similar genes with the exact same base sequence. If a color is not provided, it proves that that particular gene presented by the microarray is not present in both of the samples. Microarrays are also used to detect which genes are expressed and their level of expression. This is done by identifying the genes that are being transcribed into mRNA and the mRNA from the two types of cell would be collected, and reverse transcriptase is used to convert mRNA into cDNA. The quantity of cDNA may have to be increased by PCR, and after being denatured to give single stranded DNA and checked with microarray, the fluorescence would indicate the genes being transcribed in the cell. The intensity of the light emitted by each spot would give the level of activity of each gene. I hope you understood. Please do question number 13. If you understand. If you do not understand, please ask me questions.